The title of this talk is The Ventricular System Part 2. In this frame, you can see the brain lateral surface. The same view is here schematically represented with superimposed ventricular system. One lateral ventricle was once called the first ventricle and the other one the second. Currently, we call them the right and left lateral ventricles. The lateral ventricles communicate by way of the intervertebral foramen, also called the foramen of Monro, with the third ventricle. This ventricle looks like a dog about to eat a little fish. The third ventricle, by way of the cerebral aqueduct or aqueduct of Sylvius, communicates with the fourth ventricle. In this frame, you can see the CSF flowing, especially at the aqueduct. The main landmarks of the lateral ventricles are the frontal or anterior, occipital or posterior, and temporal or inferior horns, the trigone where they meet, and the body. The main landmarks of the third ventricle are the supraoptic, infundibular, suprapineal, and pineal recesses. In the third ventricle, we also see the massa intermedia or interthalamic adhesion. The interthalamic adhesion is a band of gray matter that connects both thalami by going through the third ventricle. The main landmarks of the fourth ventricle are the body, Pastigium, because it is reminiscent of the summit of a Greek building, the lateral recess, foramen of Luschka, and the foramen of Magendie. The obex, the area where the fourth ventricle narrows to meet the spinal central canal, is also a fourth ventricle landmark. Now we will use the lateral view of the brain to indicate the level of cut that will allow us to see ventricular-related structures. The first cut is done rather high in the cerebrum. In it, we see the lateral ventricles. They are surrounded by white matter. As you can appreciate, the choroid plexus is very prominent. Now, let's bring back the figure indicating the level of the first cut so we can appreciate the angle of the new cut. The cut at this level shows the lateral ventricle housing the choroid plexus, the anterior horn, body, trigone, and posterior horn of the lateral ventricles. At this level, the lateral ventricle boundaries consist the fibers of the genu and the splenium of the corpus callosum. The splenium of the corpus callosum connects the occipital lobes and is fundamental for vision. The head of the caudate nucleus forms the lateral boundary of the anterior horn. This next cut at the level shown shows a significant difference appearing. To start with, we see the top part of the cerebellum. The anterior horns of the lateral ventricles really look like horns. They are separated by the septum pellucidum, formed by two sheets of glia cells called lamina, separated by a narrow CSF filled space called the cavum septum pellucidum. The septum pellucidum at this level attaches to the body of the fornix, as indicated by the interrupted line in the added figure. The fornix is a bidirectional highway between the hippocampus and the diencephalon, as well as a commissure connecting both hippocampi. The posterior horn at this cut are rather small. The third ventricle can be seen and the ambient and quadrigeminal cisterns appear. The ambient and quadrigeminal cisterns are part of the perimesencephalic cisternal system. I will take a few minutes to describe this system. This new frame represents a cut at the level of the midbrain, going through the aqueduct. In it, you can see the constituent of the perimesencephalic cistern system, the quadrigeminal cistern, ambient, crura, and interpeduncular cisterns. The interpeduncular cistern is also connected to the carotid, suprasellar, and to the just added prepontine cistern. Now, we will return to the last cut used for the anatomical description of the ventricles. We are going to do so to use it as reference for the next cut. This new cut is angled down and posteriorly. In it, we see a similar ventricular configuration and a slightly more cerebellum. We see the quadrigeminal and the ambient cistern at the lower level. We also see the posterior commissure. The posterior commissure at this level forms the posterior border of the third ventricle. The posterior commissure, here highlighted in blue, is constituted by a bundle of white matter connecting the superior colliculi. The posterior commissure contributes to the symmetry of pupillary reactions, is involved in controlling vertical eye movements, and in the integration of all sensory modalities to extraocular movements. Further posterior tilting of the cut angle, as you can see in this frame, exposes a larger portion of the cerebellum and the fornix. The fornix now looks like two structures, indicating, as you can appreciate in the added figure, at the level of the interrupted line that the once united fibers are now separated, forming the columns of the fornix. The Intrathalamic adhesion is also present at this level. This new cut produces a coronal view of the brain. In it, you can see the anterior horns of the lateral ventricle under the body of the corpus callosum. The corpus callosum in this cut forms the roof of the anterior horns of the lateral ventricles. We can also see the septum pellucidum, the fornix, the third ventricle, and the foramen Monro. In this coronal specimen, you can see a yellow tube inserted through the right foramen Monro. This new specimen corresponds to a mid-sagittal cut. We can see the choroid plexus of the third ventricle, the cut interthalamic adhesion, the aqueduct, the fornix, and the foramen Monro. This lower mid-sagittal cut specimen shows the cerebellum, tonsils, inferior velum, and the region of the cerebello-medullary cistern. In this coronal view of the brainstem showing part of the left cerebellar hemisphere, we can also see the choroid plexus of the fourth ventricle, pretty well preserved on the left side and poorly preserved on the right. In the course of this talk, we have mentioned the choroid plexus at different cuts, in the lateral ventricles, third ventricle, and the fourth ventricle. So it is time to ask ourselves, what is the configuration of the choroid plexus? In this figure, we have traced the right supratentorial ventricular choroid plexus to the floor of the body of the lateral ventricle, rostral wall of the trigone, and roof of the temporal horn, to the right orifice of Monro and the roof of the third ventricle. Now we added the left supratentorial choroid plexus at the same location. The infratentorial choroid plexus occupies the region of the inferior velum and extends to the foramina of Lucia and Magenti. In order to better appreciate the configuration of the infratentorial plexus, we should start by removing the bulk of the cerebellar hemispheres to look in the direction of the cut. From this perspective, we see the dorsal brainstem, the cut superior and middle cerebellar peduncles, the floculi, and the choroid plexus covered by the inferior velum. Here, we have isolated the choroid plexus to better appreciate its anatomy. The fourth ventricle choroid plexus has a medial segment with a nodular part and a tonsillar part. The tonsillar part exits the posterior fossa through the foramen of Magenti. The medial segment is flanked by two lateral segments. The lateral segment has two parts, the pendular part and the floccular part. The latter exits the ventricle through the foramen of Luschka. So, fourth ventricle outflow takes place by the medial Magenti and the two lateral Luschka foramina. Now, I would like for us to look at the brain from above to see the distribution of the choroid plexus in all the ventricles. Active secretion by the choroid plexus is classically considered the source of most CSF, but tissue fluid from CNS parenchyma cells to the paravenular space and from the ependymal cell to the ventricles adds to its volume. So let's summarize these important points. This chart gives you an overview of the site, mechanism of CSF production and their site of release. Active secretion by the choroid plexus is classically considered the source of most CSF. Choroid plexus CSF production spills into the ventricles. The mechanism of CSF production at the choroid plexus was explained in the prior video labeled Ventricular System Part 1. Leakage from ependymal cells contribute to CSF volume. Leakage from the ependymal cells also spills into the ventricles. The third and last mechanism of CSF production is CNS parenchymal leakage. CNS parenchymal leakage spills into the paravenous space. We will describe the paravenous space and explain its relation to CSF production at the same time as we address CSF circulation and reabsorption in a few minutes. But first, we will address what are the functions of the CSF. The functions of the CSF are to provide physical protection against impact and sudden movements and immunological protection against microbes, to provide buoyancy, thus making it easier for the body to carry the brain, to provide the brain and the spine with nutrients, to be a waterway to transport hormones and a route to dispose of waste, and the last function is to be the gatekeeper, maintaining fluid balance and a stable chemical and ionic CNS environment. How are CSF functions accomplished? The physical properties of CSF explains its capacity to provide protection, buoyancy, and a circuit for hormone transportation and waste disposal. The relatively large volume of 400 to 600 milliliters in adults and the quick turnover of 3 to 4 times a day explains its buffering effect against osmotic and chemical changes. The presence of glucose and vitamins explains its nutritional value, and the presence of white cells and antibodies explains its immunological attributes. The mechanics of all these functions are important, but in this talk we will only discuss waste disposal. The CSF has an active role in waste disposal, which is closely linked to CSF production. As you will soon see, CSF active role can be compared to the cleansing effect of running water on the rocks embedded in the course of a fast river. The CSF not only circulates through the ventricle cisterns and supranuclear space, but it also flows through the CNS parenchyma. We will illustrate this later process in the next figures. I must warn you that uncertainties about CSF production, circulation, and reabsorption are still present, but I will put forward the one explanation I feel more likely to be plausible. In this figure, you can see represented an artery, the pararterial space filled with CSF. This space is also called the virtual Robin space. I am distinguishing pararterial space from periarterial space. The latter, not represented in this figure and in this talk, is reserved for the space between the smooth muscle cells forming the wall of the arteries. In this talk, we will not assign a CSF waste disposal function to this space, but some authors do, and they may be right. The area on the brackets corresponds to the CNS parenchyma. In it, we find aquaporin 4 channels represented by purple dots embedded in the astrocyte end feet, fibrillary astrocyte neuron, another fibrillary astrocyte with aquaporin 4 channels embedded in the astrocyte feet. Now I am indicating to the paravenous space filled with CSF. This space is also referred to as virtual Robin space, same as the pararterial space. The last structure indicated is the vein. The just added black arrows indicate the direction of CSF flow in the pararterial space. The thick arrow indicates flow towards the depth of the CNS parenchyma. The curved arrows indicate the flow towards the astrocytic end feet and the pressure this flow produces against the walls of the pararterial space. The added arrow in the paravenous space indicates the flow towards the supranuclear space. I have now represented the interstitial fluid as yellow dots, and now the waste product around the neurons as red dots. I have now added what I am referring to as the flowing CSF. The flowing CSF will be represented as light blue dots going through the layer of aquaporin 4 channels facing the pararterial space, as you see here. This is the beginning of the journey of CSF, which we have come to call the lymphatic system. The term lymphatic system was constructed by adding the letter G to lymphatic. The G was chosen because of the importance of glia cells, that is the astrocyte, to this system, as you will see. I am now representing the flowing of CSF in the astrocyte end feet. Now the first wave of flowing CSF appears on the channels facing the parenchyma, and now they have entered the interstitial space. The flowing CSF advances in the interstitial space, likely propelled by the force generated by the arterial pressure produced by cardiac contraction, as you see here. Now you see a trio of light blue, yellow, and red dots. This represents the flowing of CSF taking along interstitial fluid and waste. Ultimately, this trio reaches the other side and are taken up by the aquaporin 4 channels, enter the astrocytic end feet, taken up again, but this time by the aquaporin 4 channels facing the paravenous space and spilling them into the paravenous space. In this way, the flow of CSF across the parenchyma contributes to the increased volume of CSF by adding interstitial fluid and waste. The CSF in the paravenous space flows in the direction of the supranuclear space, as indicated by the arrow. Where does the paravenous CSF go? It is not clear if the paravenous space drains to the supranuclear space, the meningeal lymphatic, or both. In this cartoon, you can see an arachnoid granulation, the superior sagittal sinus between the outer and inner dura, the supranuclear space below the inner dura, and a meningeal lymphatic vessel in the dura. In this frame, I have represented a breaching vein with its paravenous space. The paravenous space are draining into the supranuclear, as you can see. I am now representing another breaching vein with its paravenous space draining into the meningeal lymphatic vessel, as you can see. It is important to know that the meningeal lymphatic vessels are present at many sites in the skull and not, as arachnogranulations are, confined to the skull area where the parasital dura is. This point is highlighted in this frame. The green vessels represent meningeal lymphatic vessels. The yellow area, the parasital dura. How CSF joins the general circulation. This chart condenses our knowledge, as I understand it to be, of CSF circulation and reabsorption. Once the CSF reaches the supranuclear space, regardless if it originated from the ventricles or from the intraparenchymal paravenous space, the CSF can follow three routes. One route is the traditional pathway, which involves the arachnoid granulations, sagittal sinus to internal jugular vein. This route will be illustrated in the next few frames. In this figure, you can see arachnoid granulations on the right hemisphere, looking like a conglomeration of small sand pebbles. The granulations bring the CSF to the sagittal sinus. From there, mixed with blood, it goes to the transverse sinus, sigmoid sinus, to reach the internal jugular vein. In addition to the classical pathway, CSF in the supranuclear space may drain via the perineural sheath surrounding cranial and spinal nerves and via the cribriform plate. From these sites to the cervical lymphatic system. From there to the lymphatic duct, subclavian artery, to reach the internal jugular vein. I will illustrate this pathway in the next frames. In this diagram we have the medulla. From it a cranial nerve is exiting, which is followed by CSF past its intraosteal portion till reaching the cervical lymphatics. This figure gives you a more panoramic view. The green represents CSF spaces, and as you can see, sleeves of green is present in all represented cranial nerves. Notice that cranial nerve 3, 4, and 6, and 12 are excluded. So all the listed cranial nerves participate in CSF clearing, as do all spinal nerves. The spinal nerve's perivascular CSF pathways to the lymphatic system is not known. Hence, we will restrict our explanation to cranial nerves with the understanding that a similar process occurs in the spinal nerves. It is interesting that cranial nerve 1 and the area of the cribriform plate is very much involved in CSF reabsorption. In this drawing, you can see the cribriform plate, an olfactory filia, and 3 lymphatic passages. Notice the 3 arrows. This one indicates a passage from the cerebral supranuclear space to a lymphatic vessel. This one from the arachnoid space around a filia directly into the lymphatic vessel. And this one indicates CSF spilling through the nasal mucosa prior to reaching the lymphatic vessel. Once CSF from the cranial nerve reaches the cervical ganglion, the CSF travels through the lymphatic duct to subclavian vein to reach the internal jugular vein, as we previously mentioned. So we have discussed the passage of perineural supranuclear CSF to the internal jugular vein. Now let's look at the last route. The CSF in the intraparenchymal paravenous space may travel in the breaching cerebral veins paravenous space to the meningeal lymphatic vessels to reach the internal jugular vein lymphatic system, which has been called the CSF canalicular system. From there to the lymphatic duct, subclavian vein, and internal jugular vein. In the next few frames, I will illustrate this system. The paravenous space, accompanied by the breaching veins, traverse the arachnoid space and the dura to reach the lymphatic vessel in the dura. In this figure, you see the intracranial lymphatic system abutting most of the cerebral sinuses. The intracranial meningeal lymphatic system drains to the lymphatic vessel linked to the internal jugular vein, going to the lymphatic duct, which empties in the subclavian vein, thus reaching the internal jugular vein. Thank you very much for your attention.